Hello, everybody. How are you?
Good, how are you?
You can hear and see me and see the slides, right?
Yes.
Thank you. Uh, We'll wait a few more minutes, see if more people come. Right now, we're only 42. With me, 43. Wow. These are dedicated. You guys are dedicated. Any questions that you have, general questions that you would like me to answer? Nothing comes to mind. What do I mean when you have to take attendance for the final exam? That means that when you come into the auditorium, you will have to show your ID card to the proctors. And the proctors will keep track of the attendance. So only the students that that are in the assembly hall, those are the only ones that I will accept the exams. And why is this? Because some students were able to take the exam outside of the assembly hall, not being supervised, as you can imagine. Unfortunately, this is very unfortunate that we have to go through all this. No, you don't need to get an app for attendance. Yeah, well, it has to be the Hunter College ID. It has to have your image on it.
Wait, I have a question.
Yes?
So I noticed that the final and also if you're making up an exam, right? They're both an hour long.
Yes.
So does that mean that there's going to be less questions overall for
No,
no?
no.
But we usually get like an hour and a half, no?
No, you never get an hour and a half. It's going to be the class is an hour and 15 minutes. And it's impossible to be an hour and 15 minutes because students have to come in, we have to set up. So it's always 60 questions, 60 minutes.
Okay, thank you.
So we didn't start late. We started at 4:09 and the previous exam was 4:08. There's always a few minutes for students to come in and settle down so that then I can unveil the password. So the exam is 1 hour, 60 questions, 60 minutes. And actually, I give always a little bit more time. So, okay. So let's move on. And today we're going to talk about the propagation of the action potential along the axon terminal and the difference between the axons that are myelinated and those that are not myelinated. Why is it that the speed of the propagation differs so much from unmyelinated axons and myelinated axons? But before that, I want to go back to last lecture. I finished very quickly. And this concept is very, very important for you to understand, which is the refractory period. So think about the axon, right? So when this is the graph, the action potential being generated on the axon. So this is a graph that represents the different stages. On the y-axis, you have the membrane potential in millivolts. The x-axis is the time in milliseconds. Notice milliseconds. So here's the resting membrane potential when the neuron is not being stimulated. Then you have stimuli arriving, and this is the area of the graph that represents the local potentials, right? Those that don't reach, the change in potential doesn't reach threshold. So that's what this part of the curve represents. Once threshold is reached, minus 55 millivolts, remember there's a lot of sodium channels right at the trigger zone. When that voltage, minus 55 millivolts, arrives there, then all those sodium channels are going to open. Sodium is going to rush in, and it's going to go very, very quickly up to plus 30. So you see, less than 1 millisecond that it takes. So this is the depolarization of the action potential, the depolarization state of the action potential. So what does it represent? It represents that the sodium channels have been activated. And around here, the potassium channels that open much slower, they start to opening up. Okay, once here at plus 30 millivolts, the inactivation gate of the sodium channel closes. So no more sodium can rush in. So now the potassium channels are active, and potassium starts going out of the axon, and the potential starts going down. The membrane potential starts going down, and this is the repolarization state. So depolarization, repolarization. And because the potassium channels take longer and slower to close, this goes down a little bit below resting membrane potential. So this is the hyperpolarization state, where it goes down to minus 80 millivolts, and then it goes back to the resting membrane potential. So what is the refractory period? So you see here that there is the absolute and the relative refractory period. So the absolute refractory period is the time in which all the activation, all the sodium channels are inactivated. So that means the inactivation gate is closing the sodium channel. So that means that if there is a stimulus arriving during this absolute refractory period, you cannot go up, you cannot go higher, you cannot generate another action potential. Why? Because the sodium channels are closed, they are inactive. The inactivation gate is closing the sodium channel. No more sodium can go in. So this is the absolute refractory period. Okay, when you start coming down here, now some of the sodium channels go back to resting stage. That means that the inactivation gate opens and the activation gate closes. So that means with the some of the channels that are able to be activated, if there's a super threshold stimulus, a stimulus that is very, very strong, then it can generate some action potential. But that's why it's called the relative refractory period, because there are already a few sodium channels that are in the resting stage and can be activated. So this is very important. And you see here, this is represented in another way, where what you see here, three curves, a red curve, a blue curve, and a green curve. The red curve represents the action potential. So the change in potential, in membrane potential. So it goes from resting, the local potential, then up 30 millivolts, then come down, hyperpolarization, and going back to resting. Now, here on this y-axis, it represents the permeability to sodium and potassium. So the sodium curve is represented here in green or blue. Let's say this is blue. So you see that when the stimulus arrives, reach the threshold, then the permeability to sodium increases, spikes up very, very quickly because all the sodium channels, voltage-gated sodium channels, are open. Then when it reaches plus 30, it starts coming down. So you see now the potassium permeability that depends on the opening and closing of the potassium channels. What is a suprathreshold signal? A suprathreshold signal, or stimulus, I should say, instead of signal, let's call it a stimulus, is a stimulus the strength of which is higher than minus 55 millivolts. And therefore, there's a few sodium channels that are already resting, and they can be reactivated by this threshold stimulus. So that means that it will work in the refractory period. So here's the potassium permeability. You see that the potassium channels start opening, and then slowly the permeability to potassium decreases. And you see here the hyperpolarization. So this indicates, signal. Yeah, signal of stimulus. I think in this case, they are interchangeable. So the difference between absolute and relative refractory period is absolute. No way, even if there's a suprathreshold signal generated by a stimulus, you cannot generate an action potential. Over here, in the relative refractory period, because as some of the sodium channels are starting to go back to resting stage, they can be reactivated. Okay. So why is this so important? It is important because it allows the action potential to travel in just one direction. Because just envision if the action potential that's going down the axon can also come up the axon. So that means that there would never be a resting stage because you'll go back and forth, back and forth, back and forth. But no, the action potential goes only from the cell body to the axon terminal. And why can't it come up? Because it's an electrical signal, right? Well, it can't go back because of the refractory period. So let's look here. This represents an axon in 3 colors. Green color is that the membrane is resting and it's excitable. So positive outside, negative inside. So green represents excitable membrane in the resting membrane potential. The red represents when the action potential starts to be propagated. So you see that sodium channels open. So the inside now is positive compared to the outside. So this is when this represents the action potential, the traveling of the action potential. Now the yellow between green and red, that's the refractory period. That's the period in which the axon can no longer respond to a stimulus. Why? Because all the voltage-gated sodium channels are closed. So it cannot go back. The action potential cannot travel back. So it can only travel in one direction. So, and it self-propagates. Why? Because as the sodium goes in, it changes the potential of the membrane that's close to it, right? So it's traveling down. You have the sodium that diffuses to the neighboring area, and therefore also induces a change in potential. So it self-propagates. So the question, if we have a super threshold signal, can the action potential flow backward? No. No, because this is absolute refractory period. And the super threshold signal would start at the beginning, not in the middle of the axon. So it self-propagates down the axon, and it only goes in one direction. So it's incredible that all these mechanisms developed in our body. Thank God, right? I mean, thank goodness. It's that this all is working well, and that it's the design and the architecture of this whole system is amazing. How it developed, how it evolved. Okay, so the action potential travels down the axon in one direction and it self-propagates. Okay, so how can you stop the action potential? First of all, the action potential is not like the graded potential. Remember, the local potential, it could increase. You go from -70 to -65. If there's a stimulus or signal that is a little stronger, it can go to -60 and so on. The action potential is all or none. Once it reaches the trigger zone, that's it. It goes, spikes up to plus 30 millivolts. So it travels at the maximum and constant strength, all or none. But we can mess around with the propagation of the action potential. So think about anesthetics. When you go to the dentist and you take Novocaine, you inject Novocaine to have local anesthesia, what does that do? Why does it stop? So the idea is to stop the feeling of pain. And the pain is transmitted through an action potential to the brain, to the pain area in the brain. So what happens, the anesthetic prevents the opening of the voltage-gated sodium channels locally at the site of injection. And the pain fibers are very small, they're very sensitive. So you use very, very low levels of this anesthetic. And this prevents the action potential from the pain fibers to be propagated. The same, for example, with toxic substances and fatigue also slow down the propagation of the action potential. And ice. Remember, if you have pain, you put ice, and that relieves in a certain way pain. Why? Because it slows down the propagation of the action potential. Okay, now I hope you understood this idea of the refractory period. Topical anesthetics, isn't that the same as local, topical, local? So here, so now let's let's discuss why is it that the action potential travels at different speeds in unmyelinated versus myelinated. What does propagation mean? Propagation means that it goes down, it propagates, it is continuous down the axon. It propagates. It goes from one area to the adjacent area to the adjacent, and all the way down the axon. It propagates. Okay, so in an unmyelinated fiber, we say that the propagation, the movement of the action potential down the axon is continuous. And you will understand what this means. However, in myelinated fibers, it's saltatory. So we're talking about the movement, the propagation of the action potential down the axon. So in an unmyelinated axon, as represented here, each area, each segment of the axon is going to be activated. You have sodium channels opening and closing, and then this activates the next area. The voltage changes, and this will activate the voltage gated sodium channels number 2 and then number 3 and so on. So you get these action potentials being generated down the axon from one point to the other, all the way down. So it's continuous. There's nothing interrupting it, right? Because the voltage at the axolemma, at the membrane of the axon, is going to change and opening consequently in serial manner the voltage gated sodium channels. So they open each neighboring area. One opens first, and 2, and then 3, and so on, all the way down. So it's a continuous propagation, a continuous movement of this electrical current down the axon. So it's like having a domino. One starts falling, and then all the other ones fall in sequence. The same if you light a wire, starts the fire at one end and it propagates, it continues down the wire. So this is the property of the movement of the propagation of the action potential down an unmyelinated axon. The conduction is slow, but it's non-decremental. What does this mean? That means that the propagation, the movement, is slow because you have to open and close sodium channels each time you move down the axon. But it's non-decremental. It never decreases in size. Why? Because it's an all or none response. Once you reach threshold, you get the maximum depolarization. Are multipolar the only ones that are continuous? No, I think, no, some bipolar are also, and some sensory neurons are also myelinated, yes. And sensory neurons, remember, they are unipolar. They are also myelinated. You'll see there's 3 different types of nerve fibers. We will go through this. So, yes, the bipolar is separated, but you will see that sensory neurons are, most of them or some of them are myelinated, and you will see, and those are unipolar. Very good questions. Okay, so the conduction in the unmyelinated is slow, but it's non-decremental. That means it doesn't decrease in voltage. Now let's go to the myelinated axon. So you see here in pink, that represents the axon, but it's covered, every 2 to 3 millimeters is covered by a Schwann cell in this case. So let's say this is a peripheral neuron. So this area is totally covered by a Schwann cell. And then you have a space in between, and here's another Schwann cell. So what is the advantage of this design? So here you have, these are the nodes of Ranvier. Remember, these are the areas of the axon that are naked. They don't have any Schwann cells. They're not myelinated. So you have action potential in this area. You have the sodium channels opening, And then the sodium ions are going to diffuse from this area all the way down here. And the sodium cannot escape because the plasma membrane is covered very, very tightly by the myelin sheath. So that means the current goes very, very fast in this area because it depends on the diffusion of the sodium ions. But it's going to decrease. Why? Because here you have a high concentration of sodium because you have the voltage gated sodium channels that allow sodium to come in. So by diffusion, sodium is going to go from high to low. So when it gets here, there's very low levels of sodium already. So the current has to be reactivated. So now you have another, the voltage sodium channels open again, more sodium comes in and reactivates the action potential. And now the sodium diffuses down the next segment that is covered by myelin. So that means that in this area of the axon that is covered by the myelin, the conduction is very, very fast because it does not depend on the opening and closing of voltage-gated ion channels. It depends on diffusion, but it is decremental. That means it's going to go from high to low, right? So here you see another way of showing this. First, let me point out that at the nodes of Ranvier, there's so many voltage-gated ion channels. There's 2,000 to 12,000. Oh. There's 2,000 to 12,000 voltage-gated ion channels per square micrometer. But in this area of the axon that is covered by myelin, there are only 25 per micrometer. So you see the big, big difference. And there's a need here to replenish sodium so that the action potential can be transmitted or propagated down this area that is covered by myelin. So the advantage here is that these segments that are myelinated, the sodium channels just rush through by diffusion. So let's see what this here shows us. Oh no.
Professor, I have a question.
Yes.
The internodes are where the myelin sheath is going to be covering the axon.
No, it's the opposite. The internodes here are the areas that don't have, because internodes, right? They're between the axon, the segments of the axons that are covered by myelin. The internodes are the same as node of Ranvier.
So where are the nodes, just the plain nodes?
The nodes would be here, the area that is covered. Is that correct? Does that make sense?
The area that's not myelinated is a node.
The node is, yeah. Oh, yes, you are correct. Of course, the area that is not covered, the node of Ranvier, that's the node, the internode would be between nodes. Yes, I'm so sorry. You're absolutely correct.
Yeah, non-myelinated is the nodes, and it also has the sodium channels.
Yes. And the area that's myelinated, that's the internode, because it's between two nodes, right? Makes sense, right? So this area that's covered by myelin is the internode, and the nodes are at each end. Okay? Clear? Yes?
Yes, thank you.
Okay, so let's see here. So here you see the Schwann cells, right? And now, here's where you have the nodes of Ranvier. And they are highly, and here are the internodes. So depolarization cannot occur here. So the sodium just rushes from one node to the other within the internode area. So no, this is... You see how the Schwann cell encircles the axon so that there's no way very, very few sodium ions can escape in the internode area. Okay. So sodium is replenished at nodes of Ranvier. Yes, that is absolutely correct. The nodes is where the sodium is replenished. So that's why we say that the current, the propagation of the action potential along the axon, if it is myelinated, saltatory, because it jumps from one node to the other. From one node, you see sodium coming in and going through here, moves through here by diffusion, and then gets here. So you can think of, if you're in a big hall and you want to have a message conveyed to the people that are in the last row. If you shout it out, they will hear very fast. But if you tell it to the people on the first row and ask those people to transmit it, to convey it to the people in the second row, in the third row, and fourth row, until it gets to the end, it takes a long time. However, if you shout it out, the people in the back row will hear it very quickly. So it's the same thing. Here, you're shouting out, you're having the action potential going from one node to the other, and it travels very fast. Okay. So the speed of propagation of the action potential, what does it depend on? We know that it's independent of the stimulus strength, because once you reach threshold, even if a stronger stimulus comes by, it's not going to increase. It does not go above plus 30 millivolts. It's an all or none response. But the speed depends on myelination, just as I explained. It also depends on temperature. If it's warmer, the sodium channels will travel faster, right? So if you apply ice and you cool off that area, the sodium ions will travel much slower, and therefore the propagation of the action potential will be slowed down. And it depends on the diameter of the fibers. So if the nerve fibers, if the axon has a large diameter, there's less resistance being applied to the movement of the sodium ions. If it's narrower, then there's more resistance to the diffusion of the sodium ions. So based on this, we classify the nerve fibers into three groups, A, B, C. So the A fibers are the ones where the action potential travels the fastest, 27 to 280 miles per hour. So they have the largest diameter, they are myelinated, they generate a quick response. So for Kelvin, here you see, or Kevin, that sensory fibers that are dedicated to conducting the action potential generated by delicate touch, pressure, or heat. Those are A fibers and motor neurons that conduct the action potentials to skeletal muscle. So A fibers are myelinated and they have the largest diameter. B fibers are in the intermediate diameter. They are still myelinated, but look at the speed of propagation, 32 miles per hour. And what are some examples of B fibers? Sensory neurons from body, from organs in the body. They don't need a quick response. And some motor neurons from the autonomic nervous system. Those conduct action potentials to smooth and cardiac muscle. And then we have the C fibers. These are the slowest. They are the smallest diameter. They are unmyelinated, and the propagation is 1 to 5 miles per hour. So some examples: sensory fibers for pain, touch, and pressure, and some of the autonomic nervous system, motor neurons. So you see that sensory fibers have representative of A, B, and C fibers, depending on what kind of stimulus is initiating the propagation, the action potential. Okay. Just like muscle fibers, there were different types. Here we have different types of neurons, nerve fibers. So how can pain feels, oh, let's see. Okay, is this why it's hard to move our fingers fast in the cold? Yes. Yes, because, you have to give the message, you have to send a message to the muscles to move, and the propagation of that action potential is slower. Does that mean it's harder to think when it's cold as well, like complex problem solving? Yes, it depends all on the temperature, right? On how cold it is. But now you know, right? So it's best to take an exam in an overheated room than in a freezing room. How can pain feel so extreme at times but goes so slowly? Well, because the stimulus is so strong that you have, and it takes time in terms of milliseconds, right? From the stimulus that initiates pain, that activates the pain fibers, for it to reach the pain center in the brain. If C fibers are so slow, how do we experience pain so fast? It's not so fast. It seems like it's fast, but you have to think in milliseconds. Remember that example that I gave you about the reflex. So you are much faster removing your hand from the flame than it takes for you to feel the heat generated by the pain generated by the heat activating the temperature receptors. So it takes longer to feel the pain than to generate a response to the stimulus that generates the pain. So it's all relative. So it will take you longer to feel the stimulus that is feel the pain than to generate a response. Okay. Your body maintains interheat to vital organs, internal heat to vital organs. Yeah, very good, Sophia. Exactly. That's why we have the hypothalamus, the thermostat. So there are ways to keep the body temperature at 37 degrees centigrade. You guys are very good. That's right. So even if you are in the North Pole, your body temperature is still at 37 degrees, right? Okay. So any more comments on this? Okay, now we move to the synapse. So we studied what goes in the cell body, and in the dendrites, what goes on in the axon. And now here you have one presynaptic neuron, and here the blue one, the lavender one is the postsynaptic neuron. And you know that they are not, here are the axon, the synaptic knobs. And if you magnify this area, you see that they are not in physical contact. There's a synaptic space here, synaptic cleft. So that means that when the action potential travels down the axon, there's a delay here, milliseconds, because you have the neurotransmitter being released. It has to diffuse across the synaptic cleft and then bind to its receptors. Now here, remember, we discussed this synapse when it was a motor neuron and a muscle fiber. That was the neuromuscular junction. Now we're talking about the synapse between two neurons. So the presynaptic and the postsynaptic. Okay, so where do these synapses form? The synapses can form between an axon and a dendrite. How would you call these? Axodendritic. Or between an axon and the cell body of the postsynaptic neuron. Body means cell body, body means soma, so that would be axosomatic. And you also have axoaxonic. That means one axon synapsing very close to the other axon terminal. In this case, here, the neurotransmitters are mostly modulatory, meaning that they can inhibit the transmission of the action potential and so on, or make it faster. So these are the types of synapses that can be formed. Oh my gosh.
These are,
yes?
all of these are between two neurons, right?
Yes. The synapse is always between two neurons. Now you can have many axons coming onto this postsynaptic neuron. Remember, I showed you that. It doesn't mean that there's always a one-to-one relationship. You can have 10 to 1, you can have 100 to 1. Most of the times is more than a 1 to 1. And you will see that there are circuits. And you will see how these circuits going from one neuron to another. For example, to the basis for memory. So you have one neuron and another neuron, then it has a collateral and it goes back. So it goes around and around and around, and that's how you can generate memory. So the action potential goes from one neuron to the other, and then it goes back to the first one and goes down back to the first one, and it continues in a cycle. And that's how you generate memory. But we will discuss this later. I wish I could show you the sky. It's beautiful. Tomorrow's going to be a good day. Anyway, okay, here we are now. So this was the structural classification. Now it's the functional classification. Okay, so there are electrical synapses. We talked about this when we refer to the smooth muscle and cardiac muscle. Remember that there are gap junctions that allow the sodium to go from one cell to the other. So this we call an electrical synapse. So that means the two cells are in close proximity and they have these channels. So that means that the sodium goes from one cell to the other. Remember, this allows for the cardiomyocytes, for example, in the atrium, to contract at the same time. Now here you have a chemical synapse, in which you have a presynaptic neuron and a postsynaptic neuron, and you have the synaptic cleft. So there's a delay here. The conduction of the electrical signal is delayed because you have to wait for the neurotransmitter to diffuse from the pre to the synaptic cleft, and then the neurotransmitter to bind to the receptors on the postsynaptic neuron. So two types of functional synapses. So the electrical synapse occur in the smooth muscle and cardiomyocytes, very rare in central nervous system. So this depends on the gap junctions. Here you have the chemical synapse that depends on the neurotransmitter. So there's that synaptic delay that I talked about, that when the neurotransmitter is released, it needs to diffuse across the synaptic cleft. Okay, so what are the events that happen for the neurotransmitter to be able to be released from the pre to the postsynaptic? So you know this already. You have a nerve impulse. That means an action potential that's traveling down the axon. When it reaches opening and closing voltage-gated sodium channels, right? Voltage-gated sodium channels. Those are the type of channels that are involved in the action potential. Now it reaches the axon, the synaptic knob. When the action potential travels down to the synaptic knob, here are voltage-gated calcium channels, which will open, and calcium will come in, will flood the axon terminal. This is the signal for the synaptic vesicles to move, fuse with the plasma membrane, and the neurotransmitter is going to be released. And then by exocytosis. And then it's going to change, bind to the receptor. And then what happens here? Remember, you can have inhibitory, you can have an IPSP, or you can have an EPSP. What happens to the postsynaptic neuron? You can have an inhibitory postsynaptic potential, or you can have an excitatory postsynaptic potential. In the case of the neuromuscular junction, it's always excitatory. The only neurotransmitter is acetylcholine. But here in the neuron-neuron communication, you can have excitation or inhibition depending on which neurotransmitter is being released and which receptors are present in the postsynaptic neuron. Okay, so how do you generate an excitatory, an EPSP? That means you generate a depolarization. That means sodium will go in the cell, and this will become positive. So it becomes depolarized. You go from minus 55 to plus 30, or let's say from minus 70 to threshold. So it's excitatory. An inhibitory, an IPSP, you have what goes in are chloride ions, which are negatively charged, or you can have potassium ions going out. So this makes the inside more negative. So this is hyperpolarization, and this generates an IPSP, an inhibitory postsynaptic neuron. So depolarization, it's excitatory. It goes in from minus 70 to the direction positive. The inhibitory goes negative to more negative. So let's put this in the graph. Let's say that you have this presynaptic neuron. Sometimes neurons can have two different types of neurotransmitters. So depending on the strength of depolarization, you can have either one neurotransmitter or both being released. So let's say that you have two presynaptic neurons synapsing onto a postsynaptic neuron. One of them generates an EPSP. The other one generates an IPSP. So if the strength are the same, let's say this goes from minus 70 to minus 60. Here it goes from minus 70 to minus 80. And the effect here will be null because you have an EPSP and an IPSP together, the same strength. So that means nothing is going to be transmitted. nothing is going to go down this postsynaptic neuron. So the postsynaptic neuron is the integrator. It integrates all the signals that come from the presynaptic neuron that synapse with it. Okay, so let's talk about different types of neurotransmitters.
Wait, Professor,
Yes,
can you go back to the last slide?
yes,
So are you saying that there are two neurons that are binding to one postsynaptic neuron? Or not binding, but
yes, yes. You can have hundreds. Remember in the last lecture, I don't know if you were here, but there was one postsynaptic neuron that was pink. And then you had many, many, many synaptic nodes synapsing with that one neuron. It's very rare that you have one-to-one relationship. The postsynaptic neuron receives signals from many presynaptic.
and you're saying they kind of cancel out if they're different?
Yes, exactly. They can be different or they can be all the same. We will cross that. I will explain that later. Okay. Okay. So different types of neurotransmitters. There's more than 100 chemicals that can act as neurotransmitters. EPSP, it's negative to less negative. Yes. Well, EPSP, that is absolutely correct. EPSP, because it's a local potential, is below threshold. The action potential goes to plus 30, but the EPSP is negative to less negative. Exactly. You're absolutely correct. Okay, so the neurotransmitters some can be excitatory, some can be inhibitory, others can be neuromodulated. Modulators, that means that they intensify or diminish the response of the excitatory or inhibitory. So look here, you have one presynaptic neuron that has two types of neurotransmitters. You see the large vesicles have one type of neurotransmitter and the smaller vesicles have another type of neurotransmitter. And depending on the frequency of stimulation, if it's very slight, only these vesicles will release the neurotransmitter. If the signal is high frequency, like here you see low frequency and high frequency, then the signal is strong, and then both neurotransmitters are released at the same time. So they can interact. They can have different effects. Suppose that this one is inhibitory. So if you have a low frequency, you only get excitation. If you have high frequency, then you get inhibition and excitation. And they either, depends which one will prevail. Okay, so let's see the different groups of chemicals that can act as neurotransmitters. Acetylcholine, you've heard enough about this. The neurons that have acetylcholine as a neurotransmitter are known as cholinergic neurons. Some have amino acids, some have biogenic amines. So these are all different chemical groups. Some can have neuropeptides like enkephalins. The opioids are endogenous opioids. Some are even gases. Nitric oxide is a neurotransmitter. And one neurotransmitter can be excitatory in a location and inhibitory in another. For example, acetylcholine can be excitatory at the neuromuscular junction, and it can be inhibitory at the heart. In the vagus, which is the 10th cranial nerve, when it's activated and releases acetylcholine onto the cardiomyocytes, that is inhibitory. It slows down. So the same neurotransmitter can be excitatory or inhibitory depending on what type of receptors are on the postsynaptic neuron. So all this Might be very confusing. But think about that any stimulus that comes upon our body, our mind, anything, is the effect is going to be translated into an electrical current. It doesn't matter if it's sound or sight or happiness or despair. All of this depends on an electrical current, right? So how can the brain decipher all these different stimuli, all these different feelings? It all depends on the type of neurotransmitter and the type of receptor, because the electrical current is going to be the same. It's sodium and potassium, boring, sodium, potassium, that carries the electrical current. So it all depends on which neurotransmitters and which receptors. That's what gives the specificity to the response to a stimulus of any type. Okay, so let's continue on with the neurotransmitters. So I show here acetylcholine amino acids, GABA, glycine. We'll go into more detail into these. GABA and glycine are inhibitory. Aspartic acid, glutamic acid are excitatory most of the time. Gases, nitric oxide, very important. Neuropeptides. Here you have enkephalin. So what is a neuropeptide? It's a peptide. What's a peptide? A peptide is made up of amino acids. So that means it's genetically determined. Here you have enkephalin and endorphin. These are opioids, our own opioids that we have. Our body makes our own opioids to relieve pain. You have here substance P. That's for pain. P for pain, substance P. And then you have cholecystokinin. This is for the gut, a peptide for the gut, a neurotransmitter in the gut. So polypeptides can also be neurotransmitters. So cholinergic, this is kind of boring because you already know that you have acetylcholine, it's being released, it binds to the acetylcholine receptor on the postsynaptic membrane. And then how is the signal stopped? Well, acetylcholine is metabolized by acetylcholinesterase. Remember myasthenia gravis, where the receptors are, it's an autoimmune disease, the receptors are destroyed, and you treat them with inhibitors for this enzyme. So acetylcholinesterase breaks down acetylcholine and therefore stops the signal. Does anyone else have an image of the brain and heart on this slide? Yes. We'll get there. So here we are, the cholinergic. Now, the parasympathetic. So the acetylcholine is excitatory, always excitatory at the neuromuscular junction. It's a nicotinic receptor on the postsynaptic. It's inhibitory at the other sides. For the parasympathetic fibers of the vagus, just what I was telling you, the cranial nerve. Oh, because the next slide is going to be the heart, okay? I haven't clicked on it yet. We're still here, sorry. The other one will cover this partially. So the acetylcholine receptor on the postsynaptic nerve, so it's going to be a muscarinic receptor. Here is a nicotinic receptor, and here is a muscarinic receptor. Here it's an ionotropic receptor. Here's a metabotropic receptor. You will see this in a minute. Just relax a little bit. Okay, so excitatory here, let's say this is a neuromuscular junction. Pretend this is a muscle fiber. So it's excitatory. Everywhere else, in other sites, it's inhibitory. So if you have the vagus nerve, cranial nerve number 10, when it's activated, it releases acetylcholine and the heart rate goes down. This is what you see now, right? So this is, I'm covering the previous slide. So here's the brain and the heart. So on the carotid artery, you have receptors for blood pressure. They're known as baroreceptors. Baro means pressure. So when the blood pressure increases, these receptors are activated and they send a signal through the glossopharyngeal nerve to the cardiovascular center in the hindbrain. This is the medulla, the area of the brain that contains the cardiovascular center. So you have a signal coming here. So this would be the sensory neuron. And you have an interneuron, and then its synapses with the vagus. The vagus is the largest cranial nerve in our body. It innervates many different organs. Here we are focusing on the heart. So you have now the vagus nerve, and so it's parasympathetic. Remember, long preganglionic, short postganglionic. And here you have acetylcholine being released. And the receptor here is going to be a metabotropic receptor. It's going to be inhibitory. We also call it muscarinic. So here, this receptor would be muscarinic. Nicotinic. Here it's going to be muscarinic. So you see the same neurotransmitter the response that it generates depends on what type of receptor is present and what type of channel is going to be open. Here, you're going to have a sodium channel. You have sodium coming in, so you have depolarization. Here, you can have a potassium or a chloride channel. So if chloride goes in, makes it more negative, so slow down, or potassium leaves, makes it more negative. So this is how it works. So excitatory, now we're still at acetylcholine. We have excitatory at the neuromuscular junction in skeletal muscle. So acetylcholine binds to its receptor. This is an ionotropic receptor. It opens up the sodium channel. So excitatory, depolarization. Now in other sites, like in the vagus and the cardiac muscle, it's going to be a metabotropic. So you have the receptor, acetylcholine binds to the receptor, then a message is sent down to the potassium channel opens, potassium goes out, and you have it more negative. You have hyperpolarization. So in this case, it's a metabotropic receptor. So acetylcholine, you see, binds to two different types of receptors. Ionotropic on the neuromuscular junction and metabotropic on, for example, the heart muscle. Okay. Professor, this slide is blocked by another image. I know. So it's like this. I did it on purpose so you can have both. Acetylcholine, the effects of both at the same time. Okay. Now here we are. Does this acetylcholine receptor on the postsynaptic neuron have a specific name? Yes, ionotropic, and it's a nicotinic receptor. If acetylcholine binds to nicotinic receptor, why doesn't it have the same effect as nicotine? It does. Why are you saying it doesn't? It does have. That's why it's called nicotinic.
Well, I mean,
that's why the acetylcholine receptor is called, this specific one is called nicotinic, because nicotine can bind to it.
Then why is nicotine bad for you, but acetylcholine is not? Well,
in cigarettes, what is bad is the paper and the smoke. It's not nicotine per se.
Okay, so nicotine itself is not a bad thing.
There you are. That's why you have those vapors, right? I'm not proposing you, but it's less harmful than a cigarette because you don't have vapor, you don't have smoke, right?
Yeah, isn't nicotine addictive though, like in and of itself?
Yes, cigarettes are addictive, yes.
But
Doesn't
acetylcholine,
mean that if it's taken in proper concentrations, it's not damaging.
but you can't get addicted to acetylcholine in the same way? Like the
No.
binding. Okay.
No, because acetylcholine is produced in the body all the time. Nicotine is an exogenous product, right? So acetylcholine, no, is not addictive. It's produced by our own body. So you have to take high amounts to be addictive. And, you know, I don't want, and it has to do with the center that is present in the brain. So it has to do also with dopamine. The addiction has to do with dopamine. So it's complex. Okay, now the amino acids. So we have some that generate EPSP, usually, okay? And that's glutamate, which is the excitatory amino acid or neurotransmitter in the cerebral cortex and brain stem. And then we have aspartate, which is the excitatory amino acid in the spinal cord. Okay, so these two, they are amino acids, glutamate and aspartate, one in the cerebral cortex and brain stem, the other one in spinal cord. Now, how about inhibitory? Inhibitory, we have GABA. GABA is an amino acid that is not incorporated into proteins. It's still an amino acid. It's gamma aminobutyric acid. And it's the inhibitory, it's the brake that is produced in the brain. In the spinal cord is glycine. So GABA, the inhibitory neurotransmitter in the brain, glycine, the inhibitory neurotransmitter in the spinal cord. Okay, so how is GABA able to inhibit the action potential? Why? Because of the generation, or let's say, how can it inhibit depolarization? Because it opens chloride channels. So if it opens chloride channels, chloride will go in and it will make it more negative. So it will go from -70, for example, to -80. So how can we take advantage of these receptors and this action of this inhibitory to treat conditions? Valium, which is a sedative, it calms you down. What does it do? It prolongs the opening of chloride channels that are receptors for GABA. So it sits there on the receptor and it facilitates the opening of the receptor when GABA is released. So it increases the inhibitory effect of GABA. So it's sedative, it calms you down. This other drug, valproic acid, is used to treat epilepsy. What happens in epilepsy? You have epileptic seizures. You have action potentials, electrical discharges, millions of neurons at the same time, which cause epileptic seizures. So again, you want to put the brake, you want to calm down. So you treat with valproic acid. What does valproic acid do? It increases the levels of GABA. How? It inhibits the enzyme that metabolizes GABA, known as GABA transaminase. So if you prevent GABA from being metabolized, now you have more GABA, and therefore you have more inhibition, you have more, you calm down. So you have these two drugs, Valium, that prolongs the opening of the chloride channels, which are receptors for GABA, and you have valproic acid that prevents GABA metabolism by inhibiting the enzyme. So you see here the GABA receptor. It is an ionotropic channel. Sometimes it can be metabotropic. In this case, what is represented here is an ionotropic. It's a chloride channel. So when GABA binds to it, chloride comes in. If it's a metabotropic, then potassium channels open and potassium is released. So the GABA receptor is a chloride channel or a potassium channel, and it generates an IPSP. Now we go to glycine. Glycine, remember, is the inhibitory neurotransmitter at the spinal cord. So you see this interneuron in the spinal cord. It's known as the Renshaw cell. So this interneuron is the neurotransmitter that it releases is glycine. So again, the GABA and glycine, they put the brakes on the action potential, on the conduction, on the propagation, or let's say on depolarization. So if you know strychnine, I don't know, maybe some of you have heard of it or not. It's a poison, very strong poison. And what it does, it binds to the glycine receptors. So that means we don't have that break, that IPSP. So you have massive tetanic contractions because there's nothing to stop the signal. So this is the story for the inhibitory neurotransmitters. And glycine is the channel is an ionotropic channel that is a chloride channel. So when glycine binds to the receptor, it opens up the channel and chloride goes in, making the inside more negative, so hyperpolarizing. Okay, now other types of neurotransmitters, biogenic amines. One type of a biogenic amine is catecholamine. Catecholamine has this specific ring, the chemical structure. Then there's three neurotransmitters that are catecholamines: norepinephrine, epinephrine, and dopamine. Norepinephrine plays a role in wake state and dreaming. It's also very important, if you remember, for the autonomic nervous system. Epinephrine, which is it's a hormone that is released by the adrenal gland. The adrenal gland is above the kidney, and this increases heart rate. So here you have one neurotransmitter that is in the parasympathetic nervous system, that is released by the vagus cranial nerve, and it slows down heartbeat, parasympathetic nervous system. On the other hand, epinephrine increases heart rate, so related to sympathetic nervous system. Now you have dopamine also. This is a very, very important neurotransmitter. It gives you the sense of pleasure. It plays an important role in addiction, all kinds of addiction, alcoholism, gambling, all kinds of addiction. Dopamine is involved. And it's present in the, it's a neurotransmitter in the substantia nigra. It plays a role in emotional and autonomic movement and in Parkinson's disease. And dopamine can be excitatory or inhibitory. All of these can be excitatory and inhibitory. So it depends on what type of receptors they bind to. So these three neurotransmitters, they all come from tyrosine. Tyrosine is an amino acid. So it's the precursor of dopamine, norepinephrine, and epinephrine. So tyrosine is converted to L-DOPA, L-DOPA to dopamine, dopamine to norepinephrine, and norepinephrine to epinephrine. One interesting aspect is that in Parkinson's disease, what happens is that the neurons in substantia nigra, they degenerate. We don't know why. Why is it that only these neurons degenerate and not the ones next to them? We don't know. These neurons contain a pigment, neuromelanin, and that's why this area is known as substantia nigra. It's the only species where these neurons in the substantia nigra contain neuromelanin. So these neurons degenerate. And because you don't have enough dopamine, the Parkinson's patients have a lot of tremors because you don't have the brake to inhibit, to control the action potential. So you could say, why don't you treat Parkinson's patients? Just give them dopamine. If the dopaminergic neurons in the substantia nigra degenerate, just give them dopamine. Well, dopamine does not cross the blood-brain barrier. So the brain has a membrane that doesn't allow things to go in. It's very hard to get across the brain, the blood-brain barrier. So usually Parkinson's patients are treated with L-DOPA that can cross the blood-brain barrier. So why can't we give GABA? What about giving GABA? Can that pass? No, it's an amino acid. It doesn't. Does it get into the brain? Guys, you have such good, I have to think about. No, you cannot treat with GABA because remember, it is an inhibitory neurotransmitter that is present all over the brain, right? So it wouldn't be a good idea to inhibit. It has to be something that is specific. Okay. So these neurotransmitters, these catecholamines, how can they be removed from the synaptic cleft to stop the signal? So they are taken up. They go back into the presynaptic neuron by transporters. So they're transporters that are specific for dopamine, others for norepinephrine, others for epinephrine. So these transporters are proteins that will actively take the neurotransmitter back into the presynaptic terminal. And here, they can be metabolized by enzymes such as catechol-O-methyltransferase, which is COMT, or monoamine oxidase. So another way, Sophia, that that Parkinson's patients are being treated with is inhibitors of this monoamine oxidase to increase the levels of dopamine so it's not destroyed, not so much is being destroyed. Okay, so final slide. Wow, today it's good. Oh, another group of biogenic amines, we have serotonin. Serotonin is a very interesting neurotransmitter. Its precursor is tryptophan, which is another amino acid. And serotonin is the mood neurotransmitter. It makes us happy. If people are depressed, they have low levels of serotonin. And some of these patients are given Prozac. So Prozac is a medicine that is antidepressant. Why? Because it prevents serotonin to be taken back into the presynaptic neuron. So that means there's more serotonin left at the synaptic cleft. So serotonin is the mood neurotransmitter. And it's released in the specific area that's known as the raphe nucleus in the brainstem. And then you have also histamine, which less is known, and it's involved in memory also. And it's also the precursor is another amino acid, histidine. So these two, histamine and serotonin, they're not catecholamines, they are indolinamines because they have a different backbone. Okay, so we will stop here today. That GABA suggestion was very interesting, but I don't think it would be very useful. Anyway, it would be kind of dangerous. Okay, so have a good weekend, and I will see you Tuesday.
Thank you,
And
Professor.
thanks for sticking around. Bye.